In this first video on the cytoskeleton and cell motility, I will discuss the major functions of the cytoskeleton of the cell and the basic components of the cytoskeleton. So the cell cytoplasm might appear to you under the light microscope as just composed of the jelly-like matter. So it's sort of empty except for the jelly-like matter. However, if we use a microscope that is powerful enough and the correct staining, as you can see here in this picture, the cytoplasm is actually quite jam-packed with materials. In this case, you can see these orange components in the cytoplasm, and they look like they're made up of rods. These are actually part of the cytoskeleton of the cell. So the cytoskeleton actually fills up a majority of the cytoplasm, and we just cannot see it under the light microscope because these components are very small, very fine. So what are the major functions of the cytoskeleton? First, they provide structural support and maintenance of cell shape. So that is quite understandable, and that's where they got their name as the skeleton of the cell, because they provide for support. Number two, they also provide framework for positioning of organelles within the cell. So without the cytoskeleton, the organelles would be all jumbled inside the cell without proper organization. So with the help of the cytoskeleton, the organelles will have a platform to stay onto and not just fall wherever gravity will call them to go. The number three, very important, the cytoskeleton also provides a railway, a track for organelle and vesicle movements. So in the previous lesson, we included in our lecture how vesicles move. So the movement of vesicles are because of motor proteins attached to the vesicles, and these motor proteins are guided in terms of the direction of the movement by the cytoskeleton itself. And number four, the cytoskeleton also enables certain cells to be motile. Okay, the structure of the cilia, the flagella, and the pseudopodia are actually very much rooted to the cytoskeletal elements of the cell. And lastly, the cytoskeleton also aids in cell division. So as we will see later on, a component of the cytoskeleton called the actin filaments is very important in cell division, as well as, of course, the microtubules that help in the separation of sister chromatids in some stages of cell division. So here we have an epithelial cell. We can see that this is a polar cell. This could be a cell on the intestine, the small intestine. Here's the portion where there are microvilli, and here's the portion at the bottom that's attached to the basement membrane. Okay, so what are the functions of the cytoskeletal elements here? Number one, structure and support. So the actin filaments that we see here actually allow these microvilli to stand erect, okay, and not to be flimsy. So actin filaments in this case are acting for support. And here we have the microtubule, this blue one. It also supports the cell to be able to stand erect in this direction. What else? We have here the intermediate filaments, okay, which also connect to the desmosomes, if you can remember our previous lesson on the ECM. And this also helps in structure and support, particularly in support. against shear forces. The number 2, intracellular transport. So we can see here that this microtubule actually is serving as a railway for this vesicle. If you can see the purple there, that's the vesicle. And if you can see the legs of the vesicle, those are the motor proteins. Okay, here's another vesicle, and here's a larger view of the motor proteins, which actually walk along the length of the microtubule. Number 3, contractility and motility. In this particular example, for the epithelial cell, there's no much need for that, and so the cytoskeletal elements do not really function for contractility and motility for epithelial cells. And what about spatial organization? These microtubules that you see here, which emerge from this portion, which is the centrosome of the cell, actually serve as an attachment for the organelles. In this case, this Golgi apparatus is spread across these 3 strands of the microtubule. So, as spatial organization. Then here we have in the nerve cell, for structure and support. We can see here also that the cytoskeletal elements serve as structure for, say, the axon of nerve cells. Then we have intracellular transport. The movement of neurotransmitters from the cell body to the synapse is actually aided by transport proteins which move along microtubules, as we see here. Then contractility and motility, particularly in this portion of end portion of the nerve cell, where it meets another nerve cell through the synapse. So these projections need to be able to be motile and contract also when needed. And for spatial organization, there is no much need here in the axon portion of the nerve cell because there's a lack of organelles here. Maybe the spatial organization will be useful in the cell body of the nerve cell. Then here in a dividing cell, number 2 is intracellular transport. Transport needs to happen here as these microtubules grow. Okay, so these microtubules serve as the tubules that attach to the chromosomes when sister chromatids are supposed to be pulled away from each other during anaphase. Then here for contractility and motility, the formation of cleavage furrow in animal cells is actually caused by the contraction of actin filaments that act like rubber bands to pinch the 2 cells off from each other. So here are the major components of the cytoskeleton. We have the microtubules, the intermediate filaments, and the actin filaments. So let's look at them one by one. So for microtubules, these are made up of heterodimer of alpha-beta tubulins, which are attached to GTP. Okay, so that's their source of energy for their polymerization. And so those are the subunits of microtubules. Take note also that there is a positive end where the beta tubulin is found, and that's where the microtubule may grow, may add more monomers in order for it to grow. And take note, this is very important, microtubules are polar, meaning to say there's a positive and a negative end. And they have an enzymatic activity, GTPase, which allows for the polymerization of the monomers or subunits of the microtubule. And then there are associated motor proteins for microtubules, the kinesins and the dyneins. All right? So those are the most important characteristics of microtubules. So the rest of these you can just read through, okay? And remember, because later on in this particular lesson, we are going to go back to these. So for example, the microtubule-associated proteins and the other structural characteristics of microtubules. And then next, we have the intermediate filaments. So unlike the microtubules, the components of intermediate filaments are very varied. There are around 70 different components. And notice here, there is no preferential site of incorporation, aside from being internal. So there's no positive or negative end. Thus, there is no polarity as well. And there is no enzymatic action. There's no GT-PACE or AT-PACE involved. And there are also no motor proteins associated with the intermediate filaments. I notice here an associated protein is plaquin, the one that you find in your desmosome and hemidesmosome. And intermediate filaments may only be found in animal cells, while the previous one, the microtubules, may be found both in plant and animal cells. And notice also this one is present in the nucleus. And we will be discussing that in the later chapters. Then actin filaments, in terms of subunits, they have actin monomers with ATP attached to them. And therefore, their enzymatic activity is as a GT-PACE to allow for the polymerization of the monomers. They have a positive and a negative end. They are polar, just like your microtubules. And therefore, they involve motor proteins called myosins. Their major associated proteins are the actin-binding proteins, and they are present in all eukaryotes as well. So, be familiar with all of these characteristics of the major components of the cytoskeleton of the cell.